The structure of DNA is different from the structure of RNA, even though they are made of almost the same materials. Both DNA and RNA are made of nucleic acids, which are long chains of nucleotides. The most visible difference is that DNA consists of a double-stranded helix, while RNA is only single-stranded. The nitrogenous bases in DNA and RNA are also very similar. 3 out of the 4 different bases are the same. The difference is that, while DNA has thymine, RNA has uracil instead. So the pairings are adenine with thymine, or adenine with uracil, and then cytosine is always paired with guanine. In DNA, C and T are pyrimidines, which means they have a single ring structure, and A and G are purines, which means they have a double ring structure. Both DNA and RNA have a sugar phosphate alternating backbone, but the difference is that DNA has a deoxyribose sugar and RNA uses ribose sugar. The first step in DNA replication is that the original DNA must untwist and replicate. The strands separate from one another and new nucleotides line up along the original DNA strands so that A pairs with T and C pairs with G. The nucleotides are attached by enzymes to form a new DNA strand. When replication is complete, each new daughter DNA has one newly made strand and one old strand from the parent DNA. The daughter DNA is identical to the parent. The origins of replication are specific sites of DNA where proteins attach themselves to separate the strands at the beginning of the process. Replication bubbles form at the origins of replication as the parental DNA separates and the daughter strands lengthen. Eventually, all of the bubbles merge, and there are 2 completed daughter strands. DNA polymerase is the enzyme that links the nucleotides of DNA to the daughter strands as it is made. The 2 sugar phosphate backbones of DNA run in opposite directions. Each backbone has a 5' end and a 3' end. Nucleotides are always added to the 3' end of the strand, and never the 5' end. One daughter strand can be made in a continuous piece by DNA polymerase. However, the other daughter strand must be made in short pieces. DNA ligase is the enzyme responsible for linking the pieces together to make a single DNA strand. Transcription is performed in the nucleus and transforms DNA into RNA. Translation occurs in the cytoplasm and turns RNA into proteins. Genetic information encrypted into DNA and RNA are similar to a foreign language. Their languages are very similar in that they are written as a linear sequence of nitrogenous bases. These sequences are highly specific and have a designated starting and ending point. They are what make up the genes on a DNA strand. Each gene can have thousands of nucleotides that make up its sequence. RNA uses codon triplets to code for a specific amino acid. These codons are 3 base combinations that become a corresponding amino acid during translation. Because the bases of RNA, A, U, C, and G, must be able to code for 20 different amino acids, a set of 3 is the smallest length that the codons can be. Actually, there are enough triplet combinations so that 2 or more codings may represent the same amino acid. For example, GAU and GAC both code for aspartic acid. However, one triplet codon cannot represent more than one amino acid. So GAU could not stand for aspartic acid sometimes, and glutamine at other times. Transcription is when the language of DNA is converted to the language of RNA, and translation is when the language of RNA is translated into the language of proteins, which is the 20 different amino acids that the codons stand for. RNA is made through the process of transcription. Firstly, one of the DNA strands acts as a template for the RNA molecules to line up next to and form hydrogen bonds with the other nucleotide bases on either side with the help of a transcription enzyme called RNA polymerase. The pairings of CG and AT still apply here, but instead of putting thymine with adenine, the RNA nucleotide uracil takes its place instead. This all starts happening when RNA polymerase attaches to a specific site on the DNA strand called the promoter, which gives the start transcribing signal. Whichever DNA strand the RNA polymerase binds to is the DNA strand that is used as the template. The 3 steps of transcription are initiation, elongation, and termination. Initiation is when the RNA polymerase enzyme connects to the promoter and RNA starts to be assembled. Elongation is when the RNA starts to veer off of the DNA template, which allows the 2 DNA strands to come back together once the RNA for that section has been made. Elongation continues until the RNA polymerase reaches a site on the DNA called the terminator, which signals for the RNA to stop being made because the gene is coming to an end. This last stage is called termination. Now that the RNA has been made, it must be processed before it can leave the nucleus. A type of RNA called messenger RNA, or mRNA, is used to encode the amino acid sequences and transfer genetic information. mRNA is made when exons of a gene are joined together. Most DNA genes are made of both introns and exons. The introns are non-coding regions, which means they have no effect on the gene and are removed before the RNA leaves the nucleus. Once the introns are removed, the remaining exons are joined together to make mRNA. This procedure of cutting and pasting introns and exons is called RNA splicing. The RNA is now ready to leave the nucleus. tRNA stands for transfer RNA, which is a special kind of RNA that acts as a molecular interpreter to translate the codons of nucleic acids into amino acid proteins. It is the tRNA's job to match amino acids to the correct codons in order to form a polypeptide, which is a chain of amino acids. Because of its structure, tRNA is able to do this by picking up an amino acid and recognizing the appropriate codons in the mRNA to which the amino acid goes. tRNA molecules consist of a single strand of RNA that has about 80 nucleotides. The tRNA coils to make several double-stranded regions where parts of the RNA can meet and pair up with the other sections. Every tRNA molecule has an anticodon, which is a single-stranded loop at one end that contains a special codon triplet. The anticodon can recognize the correct codon on the mRNA by using base pairing. On the other end of the tRNA molecule, there is a place for the amino acid to attach. The amino acids are linked to the correct tRNA by an enzyme. There are 20 versions of this enzyme, because each enzyme binds only one amino acid to all tRNA molecules that code for just that amino acid, which is known from the anticodon. This amino acid is then added to a polypeptide chain by ribosomes, which we see in the next section. Ribosomes are organelles in the cytoplasm of a cell that coordinate the functioning of mRNA and tRNA. They are responsible for the making of polypeptides. Ribosomes have 2 subunits that are both made of proteins and a type of RNA called ribosomal RNA, or rRNA. Ribosomes in eukaryotes are bigger and have a slightly different makeup, but they are similar in function to those of prokaryotes. Some prokaryotic ribosomes can be made inactive by antibiotic drugs, while eukaryotic ribosomes are unaffected, which is why antibiotics are used to treat bacterial infections. Next, we'll talk about the binding sites of ribosomes. Each ribosome has 3 binding sites, 1 for mRNA and 2 for tRNA. The 2 subunits are used to hold the tRNA and mRNA molecules together, which allows the amino acids on the tRNA to be attached to the growing polypeptide. Translation has the same 3 stages as transcription: initiation, elongation, and termination. The start of translation is triggered by a special codon called an initiation or start codon that gathers the mRNA, a tRNA with the first amino acid attached, and the 2 subunits of a ribosome. During initiation, a spot on the mRNA is chosen to begin, which is where that first tRNA molecule binds to the start codon. The start codon is always AUG, which means the anticodon of the first tRNA is always UAC. The 2 subunits of the ribosome bind to each other to make a functioning ribosome, and the first tRNA molecule fits into one of the 2 binding sites. That binding site is called the P site, and is where the polypeptide will continue to grow. The other tRNA binding site is currently empty and ready for the next tRNA that has an amino acid to deposit. After initiation, amino acids are added one at a time to form a polypeptide chain. This elongation process has 3 steps. The first is codon recognition, when the anticodon of a tRNA molecule pairs with the correct mRNA codon for the amino acid that it is carrying. This occurs in the A site of the ribosome. The next step is peptide bond formation, when the polypeptide separates from the tRNA in the P site and attaches to the amino acid on the tRNA in the A site by a peptide bond. One more amino acid has been added to the chain. The last step in elongation is translocation. The tRNA in the P site no longer has any amino acids, so it leaves the ribosome. The ribosome then moves the tRNA that was in the A site to the P site, along with it the attached polypeptide. The codon on the mRNA and the anticodon of the tRNA remain connected, and the mRNA and tRNA move together. Now the A site of the ribosome is empty again, and we can start the process all over. This process continues until a stop codon in the mRNA reaches the ribosome's A site. Stop codons, which are UAA, UAG, and UGA, do not code for amino acids. Rather, they send the signal for translation to stop. This is the termination stage of translation. The finished polypeptide in the P site detaches from the last tRNA and leaves the ribosome. The ribosome then breaks into 2 separate subunits. Okay, let's review what we've gone through so far. Transcription is when RNA is made from DNA, using a DNA template and the enzyme RNA polymerase. Translation is when RNA is translated into protein with the use of codons, transfer RNA, messenger RNA, and ribosomes. The processes of transcription and translation are how genes control cells, or how a genotype produces a phenotype on an organism.